It's a pleasure for me to be on this program with Harold Varmus. Harold was actually a student of mine at medical school. In the second year, he took a course with me, which I'm sure inspired his life of full achievements.
I was an older student.
Yeah, at P&S, Columbia University. Harold, as you know, was at the University of California from 1970 to 1993, and it was there that he did this Nobel Prize-winning work with his colleague Michael Bishop. In '93, he moved to become director of the National Institutes of Health in Maryland, and in 2000, succeeded me as president of Memorial Sloan Kettering Cancer Center. After 10 years in that job, he moved back to the National Cancer Institute for a few years. And during the past year, he has returned to New York to assume a position at Cornell Medical School as the Lewis Thomas University Professor of Medicine. So without further ado, Harold is going to talk to us about recent advances in research and implications for the prevention and treatment of cancers.
Okay, Paul, thank you very much. So my goal today is to give you a snapshot of what's happened in cancer research over the last 45 years or so. I'm framing this discussion with bookended proclamations by two important presidents, Richard Nixon and Barack Obama. I want to give you some flavor of the changes that are coming in the treatment of cancer. I'll have a few things to say about other aspects of cancer at the end. I realize most of you are here because cancer is something everyone's concerned about. There are over 500,000 deaths every year in the US from cancer. There is some good news buried in that. There's been about a 1.5% decline in age-adjusted mortality rates over the last 15 or 20 years. We are making some progress. But my focus here will be on what science has contributed to new ideas about treating cancer. And at the end, I'll outline some areas that I haven't had time to discuss that might be appropriate for questions. My goal is to give you some sense of these things over the course of about 20 or 25 minutes, and then leave the floor open. I hope not for questions about personal health problems or problems in your family, but instead some more general discussions. I don't have a medical license anymore. I don't know if Paul does, so I'm not going to give personal advice, but I hope we can generalize the discussion. So, as Paul mentioned, I've been around a long time, and when I began cancer research 45 years ago, my entry into the field was followed by some happy news. If we can have the first slide. And these slides may be hard to see. This is a picture of Richard Nixon in the East Room signing the National Cancer Act. That was an interesting bit of legislation. For one thing, it in some ways embodied one of the first efforts of successful lobbying for medical research money, thanks to the work of Mary Lasker and her colleagues. The bill did a number of things that are technical with respect to the Cancer Institute, but the non-technical aspect is interesting. Nixon believed that cancer was a disease we could conquer the way Kennedy took us to the moon. That was implausible at the time. We knew about cancers. We knew that tobacco smoking was a contributory factor. We knew that radiation was bad. We really didn't know anything about the essential feature of cancer, how a normal cell becomes an unregulated cell that grows inappropriately and spreads in the body and causes death. So we didn't really understand the basics of the disease. We had some tools for treatment. I'll come to those in just a moment. But what Nixon did was to provide a lot of additional resources to the Cancer Institute in particular, to NIH in general, that propelled a lot of excitement in the field and brought many people into the field and did ultimately, in an unrestricted way, give birth to many of the discoveries that play a role in how we've changed the perception of cancer as a disease today. Now, after the signing of that act, there were many new things that happen. I can't summarize them all in a short bit of time. We did begin to learn enough about how normal cells function and how cancer cells function to begin to understand how a normal cell becomes a cancer cell that is essentially a clone that then grows on to develop, to cause ultimately, over the course of many years, symptoms and signs and the lethal effects of cancer. Deeply embedded in this whole effort to understand how a normal cell becomes a cancer cell was the discovery of those genes that are likely to be involved as targets for mutations, changes in the sequence of DNA, changes in the way DNA is arranged on chromosomes, numbers of deletions and amplifications of DNA that contribute to the cancerous process. Most of those events occur as what we call somatic changes, changes in our cells as we grow and develop, not changes we inherit. But there are some mutations that we inherit. I'll come back to those a little bit at the very end, that confer a risk of cancer on individuals who inherit those damaged genes from their progenitors. This whole effort of understanding the connection between those genes that are damaged and cancer was greatly amplified by the Human Genome Project, which I'm sure all of you have heard about, something completed approximately in 2003 that provided the tools and the basic blueprint of the human genome for further understanding of the genetics of cancer. In the course of doing the studies that identified the genes most likely to be involved in cancer and beginning to understand what the effects of those mutations are on the proteins that genes encode, we began to develop some therapeutic tools, antibodies against bad proteins, drugs that inhibit bad proteins, and I'll illustrate some of those in a moment. Those were incredibly important steps because they fulfilled the promise of taking a genetic approach to cancer. And as the Genome Project evolved into a much broader effort to understand evolution and human and animal physiology, much better tools for rapid sequencing of the genome, not a few $1 billion to sequence the first genome, but now thousands of dollars to sequence anybody's genome became a standard for the day. And there are lots of other analytic tools involved that come from computational sciences, other tools for analyzing proteins and RNA that I won't get into here because I'm trying to be relatively non-technical, but they're incredibly important in thinking about how we implement the new wave of cancer therapeutics or diagnostics or even preventive strategies in the general public. Now, these factors, that many of which are the result of the avalanche of research that was triggered in part by Nixon's proclamation, we have a new edict from our current president. If you could play the movie, please.
The budget I sent this Congress on Monday will include a new precision medicine initiative that brings America closer to curing diseases like cancer and diabetes and gives all of us access potentially to the personalized information that we need to keep ourselves and our families healthier. You can match a blood transfusion to a blood type. That was an important discovery. What if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature? And that's the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person. First, we're going to work with the National Cancer Institute. We want to find the genetic factors that can lead to cancer. And we want to use that knowledge to develop new and more effective approaches to help people beat this disease.
So what the president's saying here is that he believes that the tools that we've developed for analyzing the genetic basis of cancer can now be used to change the way we diagnose cancer. Cancer is not one disease. It's not even a disease that we can characterize as lung cancer or breast cancer or liver cancer. It's a disease which is defined by the specific mutations that drive any single cancer. And it's that diagnostic criterion or criteria that should be used in designing new therapeutics. Now, we have traditional therapeutics that you all know about: chemotherapy, surgery, radiation. They are good. They're often curative, especially surgery, and they've been improving over the years. But what I'd like to do is to introduce to you the unconventional therapies that are emerging from our new understanding of cancer as a disease of the genome. So first, and perhaps most obviously, are the drugs that target those mutated genes and, more importantly, in most cases, the proteins that those genes encode. And the basic idea here is that the mutations frequently, not always, but frequently make an enzyme or some other kind of physiological factor work in a more forceful fashion. And what the drugs or antibodies do is inhibit that function. So the classic example, the poster child for this approach, is some drug you probably have all heard about called GLEEVEC, a drug that was discovered somewhat serendipitously almost 20 years ago, but about 15 years ago, one of the most exciting moments of my own life as a scientific administrator, to hear that when this drug was used to treat a set of patients who had a form of leukemia called chronic myeloid leukemia, a disease that occurs in about 6,000 individuals, usually in their mid-50s or 40s in the US each year, that essentially every patient who got that drug went into profound remission. And over the course of the subsequent 15 years, we've learned how to use the drug, and actually second and third line drugs of the same kind, when drug resistance is encountered, have a normal lifespan. Previously, they all died, all these patients died generally within 5 years of diagnosis. This drug sits in what we call the active site of the enzyme, and it blocks its activity. One characteristic of this disease is that virtually every patient has the same molecular abnormality, the same mutation in their chromosomes. And therefore, this drug works in almost every case of chronic myeloid leukemia. That is atypical. Let me give you one sense in which that is atypical by showing you a case of a young, 10-year-old boy who had a form of B-cell leukemia that was resistant to the currently used chemotherapeutic and bone marrow transplant efforts that are currently used for that disease. When his genes and chromosomes were examined, it was found he had an abnormality of the chromosomes that changed a different gene to the one that is involved in chronic myeloid leukemia. And when that was recognized and it was known that that other gene also made a protein responsive to GLEEVEC, this kid was treated with GLEEVEC, immediate improvement, which you can see by the normalization of the bone marrow, the change in the chromosomes. I won't go into the technical details here. And he remained in remission for a couple of years, still in remission as far as we can tell. And that is a good example of the kind of result that comes about by taking every person who does not respond to conventional therapy and asking, is there something else we can do with some of these new technologies? Now, it's not so easy, and this approach to therapy is not used on most patients yet, because we don't have all the tools that we need, and the situation is particularly complex. This is the only sort of data slide I'll show you. I hope you can see it. What's shown here is the constellation of mutations seen in a group of patients, roughly 230, who have what the pathologists would call the same disease, adenocarcinoma of the lung, the most common form of death from cancer in this country. And what is shown across the horizontal axis are the patient tumors. What's shown on the vertical axis are the names of genes. And each colored bar indicates a mutation of a different type. And what you can see, you don't need to understand the details here, the striking thing is that genes are involved, affected, mutated in this disease at different frequencies, that the kinds of mutations vary, and the combinations are totally different. Every patient has his own or her own disease. Some people call this the basis of personalized medicine. Personally, I say, my father practiced personalized medicine 50 years ago. He knew who his patients were. He wasn't just looking at data. But this is precision medicine at its highest form. So many of these patients, for example, have mutations in a gene called EGFR. You don't need to know what that means. It happens to be a gene I work on. And those patients are candidates for a set of new drugs. Tarceva or erlotinib is the most commonly used. And they almost always have profound remissions over the course of the next 1 to 2 years. But for reasons I'll come to in a moment, the remissions do not persist. One of the reasons they don't persist is that not only is the constellation of cancers of a certain pathological type very complex and diverse, but each patient's cancer is very complex because it's continuing to evolve. So if we think about the original cancer cell and then sample the cancer as it metastasizes to different sites over time, what we see by doing genomic analyses is that the cancer has a lot of different mutations, and as the cells divide, more mutations appear or become selected for at these various sites, not unlike what Darwin thought about when he looked at the evolution of species, the same kind of tree, and clearly this creates a level of complexity that's difficult to deal with because any single cancer may actually have many subclones, many components within it that make any therapeutic choice less likely to work for long periods of time. So the complexity of the situation, the continued evolution of cancers contributes to what is becoming a major problem in use of the targeted therapies we already have, namely resistance to the drug. There may be one cell in a large tumor that already has a mutation that confers resistance to the drug, and over time, that cell is going to survive. It's Darwin's original precept. Random changes, selection for what's the most likely to survive in the environment of a cancer drug. So often, resistance is due to mutations that affect the target itself. We know how to deal with that in many cases by designing and making new drugs that will work more effectively on the original target once resistance appears. And we have many examples of that. But resistance can also be a lot more complex with what are essentially bypass mechanisms, mutations in other genes, other kinds of signaling events within cells. One way to get over this, because this is a problem that you probably have heard about in the context of treating bacterial infections or treating HIV infection, is to use multiple drugs at a time or multiple kinds of therapeutic approaches at a time. And that kind of combination therapy is definitely on the drawing boards. The number of efforts is still limited because these drugs tend to be toxic, and the difficulty of developing the combination therapies using various kinds of models of cancer is still in early stages. I want to talk about two other unconventional approaches to therapy. The first is, the first is to exploit a deeper understanding we have about cancer cells than just an analysis of the genome. That is, what are the consequences physiologically of those mutational changes? And we know a lot about that, how these changes may affect the way DNA is modified by chemistry, ways in which DNA repair may work, ways in which RNA is revised during its synthesis. Paul Marks himself has developed one of the drugs that interferes with one of the modifications of DNA that is relevant to gene control. We're still in the early stages here. There are only a few drugs. Paul's is one of them that's been approved, but there are many other opportunities, I believe, and we can get into this in the discussion if there's some people who want to ask about it. But I do want to emphasize a third approach in more detail, and that is the approach that's offered by the immune system. We've known for a long time that the immune system works in a variety of ways, can in principle recognize cells that are perceived as foreign, and yet until just the last 10 or 20 years, there's been no good way to harness the immune system for the treatment of cancer. There was a bit of light shed on the issue a couple of decades ago when we began to use so-called monoclonal antibodies to treat cancers like breast cancers with antibodies like Herceptin, which many of you have heard about, that target proteins that are found abundantly on the surface of about 15 or 20% of breast cancers. That's an advance. We've used recombinant DNA technology to make antibodies that are gassed up by being fused to toxins from bacteria. And this seems like science fiction. It actually works for treatment of certain leukemias, mesothelioma, and a few other diseases. Not easy to do, but it does have virtue. Recently, there's been some discussion of new approaches that involve taking one of the arms of the immune system, the so-called T cells, and providing them with a way to recognize cancer cells in specific patients. This is laborious, not easy technology, but it has shown a lot of promise in a limited number of patients. What I'd like to emphasize, however, for this discussion is another more profound change that depends on our recognition that the immune system works not just by stepping on the gas and recognizing something that's foreign and killing it, but also operates in a yin-yang fashion with inhibitory forces that restrict the ability of the immune system to recognize cells. That's a useful thing normally, because if we recognized cells that were minimally altered, we would have so-called autoimmune diseases. It's good to have a restrictive system. But in a patient who is battling a cancer, which may have some subtle changes on its surface, restricting the so-called checkpoint of an inhibitory force in the immune system is potentially very useful. And indeed, over the course of the last 5 to 10 years, the use of antibodies that interfere with this so-called checkpoint control has proved dramatically effective in many cases, and is earning Jim Allison, who's the leader in that field and was a colleague of ours at Sloan Kettering, numerous prizes, which will someday, I suspect, take him to Stockholm. So let me just review a couple of things that we've learned about this new approach of checkpoint control. First, that while this work was done initially mainly with patients with melanoma, malignant melanoma, disease, cancer of the skin, now there are a lot of other cancers that have responded to this kind of therapy, including lung cancer and renal cancer and leukemias. And what's more important is that we don't simply talk here about a prolongation of life by several months, but persistent effects. Immune systems have memory, and they can remember what they've seen and keep up a response that lasts for years. Secondly, you might ask, what is the immune system responding to? And here there's an irony. You might have thought from some of the things I told you earlier that cancers are very hard to deal with because they have so many mutations. Indeed, some cancers have as many as 10,000 or 100,000 mutations in their genomes. In the case of immune therapy, that's a good thing, because many of these mutations, even those that don't drive the cancer but simply occurred as passengers during the evolution of the cancer, are making novel proteins that are not necessarily seen by the immune system as our own. They're seen as foreign. So the more mutations you have, it appears, the more likely you are to respond, even if those mutations are not driving the cancer. The third thing you might ask is, why am I talking about this when I'm talking about precision medicine? Well, many of us believe that in the near future, we will be able to predict which of those new foreign proteins, neoantigens, are the most likely to elicit the kind of immune response that we can boost with antibodies against the inhibitory forces. And there will be the link to the precision that many of us hope to see in the treatment of cancer with immune tools. Now, I said to you in the beginning that I was going to focus on treatment because that's the best way for me to tell you simply about how our new understanding of cancer is affecting how we control cancer. And you already appreciate that when I say treatment, I'm also talking about diagnosis, because you can't select the right treatment until you've made a diagnosis that's precise with respect to the mutations and the genes that affect them and the physiological consequences of those changes. But there are a lot of other things I haven't told you about that are incredibly important and, in many cases, being influenced by the kinds of things we're learning from cancer research. And I start with prevention. I'm often accused of being a proselytizer for new therapies, but I know that we could do a lot more with the tools we already have for prevention, and we can learn more about prevention in some of the ways that I've mentioned. So most of us believe we could probably reduce overall cancer rates, as incidence, as well as mortality, by doing what we already know is worth doing: shielding ourselves from UV sunlight, not smoking, getting enough exercise, maintaining a normal weight, not excessively drinking, using the vaccines that we have against hepatitis B virus and against human papillomavirus to protect us against the viruses that we know are causes of cancer. And yet we don't do that We can also learn more about prevention by looking at the mutational profiles I've been talking about, because those often have signatures that tell us what kind of agents were contributing to the mutations. So there's a lot to learn from the basic research on cancer cells as well. The second thing is I've talked mainly about those mutations that occur during our lifetimes, not about the mutations you inherit from your parents that may influence cancer risk. And we need to do more about risk assessment and get people in for early diagnosis based on those risks. Those risks may be a result of mutations like the ones that everybody in this room knows about that affect the BRCA1 and 2 genes, but also mutations that affect our ability to repair our DNA that contribute to some fairly common syndromes like Lynch syndrome. But a good doctor does a much better risk assessment than that. They also look at weight, at eating habits, at tobacco use, and UV exposure, because we can reduce risk in many cases. Early diagnosis is still problematic. It's a great thing for colon cancer, and I hope everybody in this room had their first colonoscopy by the age of 50, if not earlier, or have had fecal blood examinations, but there are also problems. Too many prostates taken out because of a positive PSA and biopsy, when the approach is not, what's been being responded to there is a tumor that would never harm the individual. But some do harm the individual, so it's hard to make those assessments. A major problem is cost. I would dispute the idea that molecular tests should be inhibitory in the cost equation. And I say that, first of all, because most molecular tests cost a lot less than on a common imaging test, of which we do many hundreds on patients at our major institutions. Secondly, the French government, which does run its medical system in a great way, has done the experiment. They've said, is it cost effective to test lung cancer patients for mutations against which we have drugs? And yes, it's very cost effective. First of all, you don't give a drug that won't be useful to somebody who doesn't have the right mutation. And secondly, you get the right drug to the right people at the right time, as President Obama suggested. But the cost of new drugs are astronomical. I don't know the easy way to correct that. The industry needs financing to develop these drugs, true, but there is an issue here. And that drives an issue that Paul asked me to at least mention, that is access to all these new technologies, many of which are not available at your local general hospital, and some only available at major cancer centers. And that is an issue of education of both patients and doctors that needs to occur. I am frequently speaking about cancer in the US because that's where most of these effects are being felt, but the NCI has, during my time there, invested a lot of money in and generated a fair amount of excitement about dealing with the rising cancer rates in the developing world. People are living longer. The increased number of cancer deaths per year, which is going to go from 7.5 million this year to about 12 million over the next 5 to 10 years, is largely going to happen. That increase is largely going to happen in the developing countries, where life expectancy is dramatically increasing, where smoking is uncontrolled. We haven't done a totally perfect job in this country. We've brought tobacco use rates down from a little over 40 to a little under 20%. That means that 1 person in 5 in this country still smokes. How crazy can you be? In the developing world, there are countries like Indonesia and Turkey and China where smoking rates are 50 to 70%. And the estimates are that we'll have roughly 1 billion deaths from tobacco alone, not all from cancer, also from cardiovascular disease and other things over the next century. And we need to get control of tobacco. But there are a lot of other things that can be done in the developing world, including, ultimately, I believe, bringing some of these new drugs there. You're seeing arguments about intellectual property over GLEEVEC, for example, already. And finally, there's a big issue here about all the information that we could get and aren't getting. Genetic analyses that are done by private companies that sequester the data, clinical data that's not shared even among hospitals, let alone put into a large database. As the Precision Medicine Initiative was originally conceived by the Institute of Medicine, the central part of it was an information database, the Cancer Knowledge Network. And that has information coming in from lots of different sources. This doesn't exist yet. It's certainly the NCI's goal to develop it, but there are lots of inhibitory forces that range from the way in which hospitals use electronic medical records to reluctance of scientists to provide data to incompatible IT systems. There are new organizations, most important of which is the Global Alliance for Genomics and Health, that are trying to solve some of these problems. But there are a lot of things on the list, and getting patients to think of themselves as cancer information donors, and doctors and scientists to recognize that we're only going to solve this incredibly complex problem if we merge a ton of data about genomes, physiology, outcomes, and hereditary factors into one database that can be appropriately mined. So we've got a lot of things to do. Right now, the next thing we have to do is for me to sit down, take some questions from you, possibly from Paul, and then the roughly 20 minutes that are left. Thanks very much.
Thank you very much, Harold. I just want to mention that in addition to the excellent summary Harold just gave us of where we stand in advancing the therapy of cancers with drugs and immunological techniques, the saying among most oncologists is, if you can cut it out, that's the best way to get it cured. So surgery and radiotherapy remain important aspects of our therapeutic armamentarium. Unfortunately, only about 30 to 40% of cancers present at an institution such as ours, Memorial Sloan Kettering, at a time when surgery is a possibility. In other words, as Harold emphasized, early diagnosis is an extremely important element in our effort to control and cure cancers. I think at this point, we have about 15 minutes left. I'd like to open it to the audience for questions for Harold. Yes, sir. Please stand. Yeah.
Is there a microphone?
Yeah.
No, thanks.
We can't hear you.
There are so many cancer therapies. Let's say that I wanted to contribute $1,000 to research in this area as you've described it. There are so many places, and I don't have a great degree of confidence as to how to get the best bang for a charitable dollar. Yeah. I know it's a digression from where you're going.
No, it's an important question because people do want to contribute, and you know, we haven't talked about NIH finances, but there's no doubt that the NIH is underfinanced at the moment. And let me just make a little digression of my own about that so that everyone understands that in the late 90s, early 2000s, the NIH underwent a doubling of resource of budget from about 13.5 to $27 billion a year. Since then, the budget has been flat and declining. Actually, over my 5 years at the NCI, our budget went down by $177 million in just real dollars, not even accounting for inflation. So because science has gotten more expensive, because more people went into science during the doubling of the budget, we have an era of dramatic hyper competition for grants, for other kinds of support, for faculty positions, for places in esteemed journals, that is forbidding a lot of important work from being done by people who are well trained. Secondly, creating a kind of dispiriting atmosphere in science, which is worrisome. So additional resources from industry, from the private sector, from donors, very important. Institutions are having to put up more money to support their research efforts at all the leading academic centers. This is a crisis that is often underappreciated because people see the NIH budget is $30 billion, what more do they need? Not true. So how do you decide to give to a certain place? I have to say that I would say, first of all, that you can go with your instinct to a certain extent. That is, if you feel you really care so much about a certain disease that I want the money to go to that disease, there's probably an advocacy group for you. Secondly, I would ask colleagues who know individuals in the academic sector to ask what advocacy groups are spending their money wisely. Most scientists know that. I'm not going to advertise for any particular advocacy group, but I can tell you that some of them are really good about getting money to young investigators, having peer review about proposals, not giving money to their favorite doctor, not spending too much money on advertising and on corporate salaries for their administrators, and you can figure that kind of stuff out with some help. There's a new organization called the Science Philanthropic Alliance, which is devoted in particular to getting money from major foundations and from wealthy people into basic research, which is a phase of research that I think is still of enormous importance, but is underfunded at the moment. And one of the things that organization is trying to do is to develop some rules for how individuals can evaluate the likelihood that the money they spend by giving it to a charitable organization is going to get to the right place. Yeah.
Yes, ma'am.
I'd like to understand.
Microphone.
I'd like to understand.
I can hear you, but they can't in the back.
Thank you so much. Thank you very much for your presentation. I don't understand why vaccines were together with obesity and tobacco under prevention.
Okay, so
Thank
let
you.
me clarify that. So what I meant by vaccines were the few vaccines we have against viruses that we know cause cancer. I've been talking a lot about genes, but the human papillomaviruses, for example, contain some types that we know are responsible for virtually all cervical cancer, some anal cancer, a lot of oropharyngeal cancer, and a few others. And those viruses carry genes that act very much the way mutant cellular genes act in causing dysregulation of growth of cells. Individuals who are immunized against the human papillomavirus vaccine do not get infected by the most highly cancer-causing strains of that virus. That vaccine is still relatively poorly used, even in the US. About half of girls and a very, very small percentage of boys who also should get the vaccine actually get all 3 recommended doses. And actually about a third get all 3 doses, about half get 1 dose. There are countries like Rwanda which get assistance and have a strong health system in which 80 to 90% of susceptible people get vaccinated. So we can do a lot better in using those vaccines. The hepatitis B vaccine is very widely used. The one deficiency there is that newborns are not getting the vaccine in parts of Africa, and that's unfortunate because a lot of the transmission of the virus occurs at birth.
Yes, sir. Peter.
Paul's calling the shot.
Peter.
Okay.
Thank you. Fascinating presentation. To elaborate a little further on the communication within the research world, big data, mathematical computation, all of the new tools of science are playing an enormous role now in the ability to match genes with specific types of cancer, et cetera, and in other areas of disease as well. How much progress is taking place in the communication among academic medical centers in terms of exchanging information and moving towards that central chart that you gave, that central place where people are not as proprietary as they
Right. Well,
have
thank you.
been in
Thank
the
you for that
past?
question. That's an
Yeah.
important question.
Before Harold answers, this has been one of Harold's major focuses over the years, and he's created several new instruments to try and achieve those goals. Sorry.
Yeah, well, in the publication domain, Paul's right. We've created a repository for published articles at NIH called PubMed Central, which is used by hundreds of thousands of people every day. I've helped to develop open access journals that get results to people immediately and change the business plan for journals. And I think we're pretty good at sharing published results. There are two problems, though. One is the problem of getting information out more quickly. Right now, it can take as much as a year or two to get a paper into the prestigious journal you need to publish in order to get your next grant or to get a promotion or get appointed at a medical center. That's a problem. And we're working on a new scheme to try to persuade medical scientists to put their work out as preprints before it's accepted for publication, to change publication practices so that the digested work that's written up gets into the public domain and into the scientific community more quickly. But to get to your question about data, we've got a big problem. In the genomics community itself, where people do sequencing, a very useful precedent was established with the Human Genome Project, that within a fairly short period of time, if you do sequencing for the Genome Project, the information goes into a centralized database at the NIH and everyone has access to it. And that's wonderful. And that has influenced the development of similar tools and similar attitudes towards sharing of information in the cancer world. But what we need at this point is not just information about genes. We need to connect those with what I call the phenotype, that is the behavior of the cancer, the response to therapy, the way the patient succeeds or fails certain therapies, and that conglomeration of information is much less easily shared. One reason that I mentioned earlier is that people use different IT systems, so there are technical difficulties that can be overcome now with so-called APIs that allow merging of databases built with different tools. Some centers are developing consortia to try to share their data, but sometimes that's done in collaboration with a private partner. And that can create difficulties because the private partner wants to have access to that data, especially once it's been massaged, for what could be, in my view, inordinate amounts of time. Ironically, virtually all the institutions that are engaged in these activities have also signed up to work with the Global Alliance for Genomics and Health, which is pledged to a much greater sharing of information, especially in the areas that it emphasizes, which are hereditary diseases, infectious diseases, and cancer. So there is a tension there, and I feel that tension every day of my life. I'm working not only with Cornell, but also with the New York Genome Center, which is a major new enterprise, which is helping to aggregate information. My goal there is to see that every individual in New York, whether they're a patient at Sloan Kettering or a patient at Kings County Hospital in Brooklyn, has access to some of these new genetic tools that may be a matter of life and death. Do you get the right therapy or not? And to do that, one of the things I hope is that all cooperating institutions are willing to get patients consented for contribution of the information about their genome and their response to therapy that will build a catalog of information that we can then mine so that we can advance this field more quickly. If we have a lot of little pieces of data aggregated in different institutions and held on to, we're not going to get this confluence of information that really allows us to lay out for the clinicians who are not doing the research what their response should be to certain kinds of genetic profiling of tumors. So, Paul.
Yes.
How do we, if we are a patient, how can we kind of force our information into the system?
That is an excellent question. The first step, of course, is being sure the information about your tumor is being obtained. And many physicians, not even oncologists, have not yet gotten into the habit of saying that every cancer ought to be subjected to at least some genetic profile, not sequencing the whole genome or even all of the coding domains, but at least a select part of the genome for which they're what we call actionable, where a mutation, if found, could change the therapy. But the next step is an important one because the doctor may say, well, okay, I've got this information, and I know you want to be an information donor, but I don't know where to put it. That's a problem. The NCI is developing a cancer genome platform that will be announced sometime in the next 6 months. And that will be a place where that information can go, but it's not going to go directly there. It'll go through intermediaries, and it depends a lot on where you're being treated. places like Sloan Kettering and MD Anderson and Dana-Farber already have databases. They're building them in the anticipation of the time when there will be a consolidated place to place them. But in other places, it's not as easy. So I appreciate the question.
Yes, sir.
Hi.
Dr. Varmus, besides tobacco and other environmental factors that you mentioned, what do you suspect may be some of the environmental causes of the mutations that, and besides long life, which of course is a great thing, what are some of the other environmental causes?
I'm glad you added long life, because that suggests that you are also thinking, as I need to emphasize now, that not all mutations that we have are attributable to some environmental issue. I mentioned in passing that when you look at the genetic profile of a cancer, we see what we call mutational signatures, not just the genes that are affected, but the actual changes that have occurred. So a C is changed to a T, or an A to a G. And when you look at a large number of those mutations in any single cancer, and frequently there are thousands, if not tens of thousands of such changes, you can begin to say that's probably the consequence of the carcinogens in tobacco smoke, or that signature is a result of an atypical enzyme that is involved in DNA repair. And we know that every time a cell divides, and your cells divide trillions of times in the course of your lifetime, it has to copy 3 billion base pairs of information and segregate 23 pairs of chromosomes to the daughter cells. Those are difficult procedures. There are corrective mechanisms. Some cells will die if they screw up, but a lot of cells get through with mutations that may have no consequence, but nevertheless are the result of the elaborate dance that cells have to go through to copy their DNA and segregate it into daughter cells. So long life is one way to prolong your susceptibility to those mutations. But there are other things that were not on my list, asbestos and other. There are some environmental exposures.
And our technologies?
They're very difficult. People talk about food. Very, very difficult to get a clear picture from dietary habits of what foods might be contributory to these underlying efforts.
And are there molecular or other types of tests that are being developed to evaluate the mutagenic potential of various
Well, it's
chemicals?
still very early stages. The folks at the Wellcome Trust Center, the so-called Sanger Center in England, have been in the forefront of developing these signatures. But it's been known for some time that the mold aflatoxin is a mutagen that causes certain kinds of mutations. They're seen in liver cancer. The tobacco signature has been known for some time. So yes, people do recognize those. Usually you can find the tobacco signature much more easily by saying, do you smoke? Although not everybody admits it.
One last question we have time for.
So I found very interesting when Google separated and became Alphabet, one of the businesses of Google, which they focused on, was this business that Dr. Levinson is sort of running for them. Yeah. And if any place has the capability of sort of the data analysis and data aggregation, maybe you can help us understand, what are they trying to do? Are they trying to sort of be the repository for all of this information and then make a ton of money? I'm just curious.
The Calico is not the place to focus here. That's an aging, a company that's devoted to aging. Cancer is probably part of the formula, but they're not going to spend much time on cancer. But Google is very, very interested in the kinds of problems that we talked about a moment ago, aggregating data. Right. And yes, they're playing heavily. Google comes to the meetings of the Global Alliance. They're competing for awards. They're actively soliciting partners to build databases. No, there's no doubt that Google will play a role here, and other companies as well, just as they have in the development of machines for sequencing, and not Google, but other companies. So the industrial component here, especially the computational folks who have recognized early on the importance of cloud computing are going to play a very significant role without doubt. And I think the goal for us is to be sure that while we recognize the commercial incentives, that we don't end up with this information locked away and separated from the other parts of the universe that need to be brought together to create an encyclopedia of what cancer is all about.
Unfortunately, our time is up. We've come a long way, obviously, in diagnosis and treating cancer. Unfortunately, we still have a long way to go. And I doubt that we'll ever be totally free of cancer. It's the nature of the disease that as long as cells divide, they're going to make mistakes, and some of those mistakes are going to become cancers. But on the good side is that we've made so much progress in our ability to detect and control cancer. I want to thank Harold for this really great